Welcome, everyone. I'm genuinely so thrilled you are joining us today for this deep dive.
Yeah, we have a really incredible topic lined up for you.
We do. But I actually want to start by asking you a question. And
Okay.
I want you to really deeply think about this as we get going today. Have you ever looked at a family trait?
Oh, this is a great question.
Right. Like maybe it's a quirky habit you share with your grandfather or a very specific natural talent your mother has, or perhaps even a deep-seated heavy anxiety that just seems to run in the family.
Yeah, the heavy stuff too.
Exactly. And have you ever wondered, am I just wired this way? Like, is my destiny completely permanently pre-written in my DNA?
It is arguably the ultimate question of human existence, isn't it?
Yeah.
I mean, are we the independent authors of our own lives making free choices, or are we just reading a biological script that was handed to us the exact moment we were conceived,
Yeah, the classic nature versus nurture debate.
but taken to an atomic level?
Okay, let's unpack this because today we are venturing into the incredible, highly flexible, and honestly sometimes highly controversial world of epigenetics.
It's a wild world.
It really is. We are going to explore this fascinating, messy space where your hard-coded genetics actually meet the unpredictable reality of your daily environment.
And we have an incredibly rich, dense source to guide us through this landscape today. It is definitely not your standard textbook reading.
No, not at all. Today's source material is an extensive, deeply detailed Dutch interview from the Wisdom podcast.
That's such a great show.
It is. The show is hosted by Sanaya Orki, and her guest for this particular conversation is Maarten Fornerod. Now, he's a retired biologist and researcher, and what is so striking about this source, right from the very first minute, is the contrasting but totally complementary vibes of these two speakers.
Oh, absolutely. It's a collision of two completely different worlds.
Right. I mean, to set the scene for you, you have the original host, Sane, who brings a very holistic, alternative health and spiritually curious perspective to the table. She is looking at human biology through this lens of overall wellness and cosmic connection.
He definitely has that modern spiritual energy.
She literally starts her show by promoting natural tree resin incense. Specifically, she talks about burning frankincense and black and red benzoin to clear the energy of the room before they even start talking.
Which is quite the intro for a science interview.
And she strongly advocates for wearing blue light blocking glasses to improve your circadian rhythm, your sleep, and your mental focus. She is very much rooted in that modern wellness mystical space.
And then, sitting right across from her on a studio, poof, you have Maarten. He brings decades of hard, rigorous, institutional scientific background.
Very rigorous.
We are not talking about a casual enthusiast here. We are talking about 14 years of intense research at the Erasmus Medical Center in the Netherlands, 10 years as a group leader at the Netherlands Cancer Institute.
Plus major stints in Heidelberg and Memphis.
Exactly. He has been studying the physical structure of DNA since the 1980s. But the kicker here is, mixed in with all that elite institutional pedigree, is this modern, almost rebellious curiosity. He is essentially a high-level biohacker now.
He's living his best life, too. He calls his current state his pre-pension, which translates to his fun retirement.
I love that concept.
Same. He actually rates his current life a 10 out of 10, which is just an amazing place to be mentally. So, what is the core mission of our deep dive today? Our mission is to explore the fundamental difference between the hard, unyielding code of our DNA and the incredibly flexible landscape of our epigenetics.
We're going to test the absolute biological limits of what we actually inherit from our ancestors.
And later on, we are going to critically examine some highly controversial claims he makes about modern medicine, biological interventions, and the pharmaceutical industry.
And my goal today, as we synthesize all this dense information, is to help separate biological dogma from cutting-edge scientific discovery. We want to help you understand just how much control you actually have over your own biology. What's fascinating here is that the science of epigenetics fundamentally shifts the narrative.
It really does.
It moves us away from being passive victims of our genetic inheritance and turns us into active participants in our biological destiny.
That is a massive shift in perspective. So let's establish the foundation of what they discussed. Let's look at the landscape of you. What exactly is the scientific difference between DNA and epigenetics? Because people throw these buzzwords around constantly, often interchangeably, and usually incorrectly.
They do, all the time. To truly grasp epigenetics, you first have to understand the baseline of DNA itself, without oversimplifying it too much.
Right.
The source explains DNA as a hard, dry code. We all know the classic image of the double helix, Fundamentally, your DNA contains the blueprint, the finite possibilities of an organism.
All the potential.
Exactly. It codes for all the tiny proteins, the gears, and the building blocks that make up your cells, which in turn make up your organs, your tissues, and everything else. It is a highly complex, incredibly stable, fractal self-organizing system.
And Maarten makes a crucial point that we have to remember. That hard code, that 99.999% of your DNA sequence, cannot easily be changed by your lifestyle.
And honestly, you really don't want it to change.
No, definitely not. If your fundamental DNA sequence is randomly mutating, that usually means radiation damage or cancer. But here is the brilliant analogy he uses in the interview to explain the difference. He says, if DNA is that hard, dry, unchangeable code, epigenetics is the physical landscape that the code lives in.
It's a beautiful, highly functional visual. Imagine the DNA double helix, not just floating in an empty void inside the cell, but resting on a physical, three-dimensional terrain.
Right. He says that some of your DNA lives in what he calls a desert. Because it's in the desert, it is tightly compacted, folded up tight, and completely inaccessible to the machinery of the cell.
So it's basically turned off.
Exactly. It's silence. It's not doing anything. But other parts of your DNA live in grassy meadows. They are open, unspooled, easily accessible, and because they are exposed, they are turned on and highly active.
And we need to graduate from the analogy to the actual biology for a second because the mechanism is incredible. The prefix epi literally means on top of or in addition to.
Oy.
Epigenetics is the layer of chemical tags, specifically things called methyl groups and specialized proteins called histones that sit quite literally on top of the DNA strand. Think of histones as tiny spools that the DNA wraps around.
Like thread on a spool.
Exactly like that. If the DNA is wrapped tightly around the spool, it's in the desert. It's silenced. If the epigenetic tags tell the spool to loosen up, the DNA unwinds, entering the grassy meadow, and the gene can be read and expressed. And unlike the hard code of the DNA sequence itself, this entire epigenetic landscape is remarkably fluidly flexible.
But wait, if the DNA doesn't change, what causes those tiny spools to wind or unwind? What's moving the DNA from the desert to the meadow?
That is where the environment comes in. In the source, Maarten describes environmental factors acting like someone walking into that microscopic DNA desert with a watering can.
Yes, he uses the Dutch word gieter.
A gieter, yes. A sudden change in your physical or emotional environment acts as the water. It can instantly trigger chemical cascades that attach or remove those methyl groups, fundamentally altering how tightly the DNA is spooled.
It just happens like that.
It can turn a genetic desert into a lush, grassy meadow in a matter of minutes or hours.
The example he gives is so incredibly relatable. Imagine you go outside on a bright summer day and the sunlight hits your bare skin. That external environmental trigger, the ultraviolet radiation from the sun, causes an immediate epigenetic change inside your skin cells.
The landscape actually shifts.
Yes, the DNA that contains the code for pigment, which was tightly packed away in the desert, is suddenly unspooled into the grassy meadow. It becomes accessible, and your cells start reading that code, producing melanin to tan your skin and protect you from further radiation.
The hard genetic code for the pigment was always there, but the environment had to turn it on.
And it is absolutely not just sunlight, it's every single thing you interact with. It's the macronutrients in the food you eat, the chronic stress you are under at work, the specific amount of deep REM sleep you get, the microtoxins you breathe in traffic.
The source notes that humans have roughly 20,000 genes, but we likely have 20,000 distinct environmental influences hitting us at any given moment. And they are all interacting in a massive, infinitely complex, real-time biological web.
So when people say to you, well, that's just the way I am, it's just my character, my dad was angry, so I'm angry, I was just born this way, the source suggests they are fundamentally missing half the picture.
Precisely. Maarten estimates that for a vast majority of complex human traits, it is a 50-50 split between your hard genetics and your environment, or perhaps 30-70, depending on the specific trait being measured. Take something as simple as hair color or height. You might have the hard genetic code for blonde hair, but if you spend all summer in the sun, your environment makes it significantly blonder. You might have the genetic potential to be 6 feet tall, but if you experience severe childhood malnutrition and environmental factor, your epigenetic landscape will adapt to conserve resources, and you might only grow to be 5'6".
The genes provided the potential ceiling, but the environment dictated the actual outcome.
Exactly.
It's a constant, lifelong negotiation between what you were given and what you were experiencing. And this brings us to a massive, deeply personal realization for you, the listener. You are an active, ongoing participant in your genetic expression.
Which is huge.
The choices you make today, what you eat for lunch, whether you take a walk, how you manage a stressful email, are constantly actively reshaping that microscopic landscape, turning thousands of genes on and off.
It is an incredibly empowering realization, but it also carries a profound weight of personal responsibility. If we accept that our environment shapes our biology so drastically, we have to look at the environments we create for ourselves.
Which is a perfect pivot into one of the most hotly debated topics in modern psychology, biology, and definitely on social media right now. And it's something the original host, Sani, was very keen to ask Maarten about.
Yes, the concept of intergenerational trauma.
Exactly. This is where the source really aggressively challenges a deeply popular cultural narrative. Sani brings up this concept she heard at a lecture at Greenwich University in London. The idea is that epigenetics scientifically proves that we inherit the specific traumas, the emotional pain, the visceral memories, and even the unique talents of our distant ancestors directly through our DNA.
It is a very romantic, very poetic, and sometimes very heavy psychological idea.
Very heavy.
The concept of ancestral trauma being physically passed down through the unbroken genetic line. It's the idea that if your great-grandfather experienced the horrors of a war, or your great-grandmother survived a famine, you carry their specific environmentally induced anxiety physically encoded in your biology today.
Right. It's everywhere in pop psychology. People use it to explain their own unexplained phobias or deep-seated depressive episodes. But Maarten fundamentally and unequivocally disagrees with this notion when it comes to human biology.
He does.
Yeah.
And he doesn't just dismiss it. He provides a rigorous biological explanation for why he disagrees. And honestly, it is one of the most fascinating mechanisms discussed in this entire deep dive.
I thought so too.
He explains that mammals, which obviously includes us humans, have a built-in, highly evolved evolutionary protective mechanism specifically designed to prevent exactly this kind of generational transfer from happening.
The wiping rounds.
Exactly. When a human body creates germ cells, meaning the sperm in males and the egg in females, there are two distinct, incredibly thorough, microscopic wiping rounds that occur during their development. The body essentially takes a massive biological eraser and systematically scrubs the epigenetic code clean.
He estimates that 95% to 99% of all that accumulated epigenetic information, all those environmental tags, the trauma markers, the chemical scars of life experiences of the parent, is completely scrubbed clean.
It's completely deleted.
It is wiped away long before the next generation is ever conceived. But wait, I have to ask. If 99% is wiped, what about that remaining 1%? Is that where this whole intergenerational trauma theory gets its footing?
That's a brilliant question. Yes, there are some tiny specific regions of the genome called imprinted genes that escape this wipe, but they are generally related to basic growth and placental development, not complex psychological trauma or behavioral memories. If we connect this to the bigger picture, you have to ask yourself, from an evolutionary standpoint, why would nature design such a rigorous, energy-intensive wiping mechanism for mammals? Maarten's answer is profound. It is specifically so that the offspring, the vulnerable next generation, is not immediately crushed by the accumulated epigenetic baggage of the previous generations.
That makes a lot of sense.
Think about it. If we physically inherited every single trauma, every starvation period, every plague, and every predator stressor our ancestors ever faced over the last 100,000 years, the biological burden on a newborn baby's nervous system would be completely unsustainable. We would be born paralyzed by genetic fear. The wipe ensures we start with a relatively clean slate, ready to adapt to our current environment, not our great-grandfather's environment.
It is literally like formatting a hard drive before you install a new operating system. You don't want all the glitchy, corrupted, malware-infected files from the last 10 years messing up the brand new computer. You want a fresh install.
That is a very apt analogy. Though, interestingly, Maarten notes a massive, crucial exception in the natural world. Plants do not wipe their epigenetic code.
I found that detail absolutely mind-blowing.
It is. Because a tree cannot run away from a threat, it can't flee a drought or hide from a swarm of locusts, it relies entirely on rapid biochemical adaptation. So, if a tree survives a severe 10-year drought, it will pass those specific epigenetic drought survival tags directly to its seeds. The next generation of trees is born already primed to conserve water. But humans, and mammals in general, we are mobile. We change our environments. So we wipe the slate.
Okay, but this raises a huge flag for me. If the code is wiped, how do we explain all the very real studies showing that children of trauma survivors often exhibit severe stress responses? Here's where it gets really interesting, and where Maarten is very careful to clarify the nuance. Saying the ancestral code is wiped does absolutely not mean parents cannot pass down trauma to their children.
Correct. The distinction he makes is vital. He makes a massive separation between ancestral trauma passed through germline DNA, which he says is wiped, and trauma experienced in real time. He explains that if a mother experiences severe acute trauma while she is pregnant, that directly and profoundly alters the unborn baby's DNA expression.
So it's not a memory being passed down, it's a real-time exposure.
Exactly. That is a direct environmental influence. The fetus is growing inside the womb. The mother's body is the entire universe for that fetus. If the mother is terrified, starving, or stressed, her bloodstream is flooded with cortisol and adrenaline. That chemical environment is the fetus's direct environment. The developing baby is being constantly hit with that watering can of stress hormones.
So the child might be born with an epigenetic landscape already primed for high anxiety, hypervigilance, or metabolic hoarding, but it is strictly because they physically, chemically experience the environment of that stress in utero. It's not because they inherited a mistinal memory from a trauma that happened decades before they were conceived.
And Maarten points out other very tangible ways traits and traumas are passed down that don't rely on epigenetic magic. Addiction, for example, can absolutely be passed down via the hard genetics. If a parent is a severe alcoholic, the child may inherit a hard genetic sequence that alters their dopamine receptors, making them highly susceptible to addiction. That is the hard, unchangeable code of the DNA sequence itself, not the flexible epigenetic layer.
And then, of course, there is the most common, most powerful way trauma is passed down from generation to generation, culturally and environmentally. If you grow up in a household with a deeply traumatized parent, their daily behavior, the chaotic or cold environment they create, the way they model reacting to stress, that becomes your childhood environment. That real-time environment shapes your developing epigenetic landscape every single day as you grow.
The transmission of the trauma is undeniably real, and the psychological effects are very real. But Maarten's point is that the mechanism is direct environmental exposure and behavioral modeling, not a ghostly memory permanently encoded in your sperm or egg cells.
It really shifts how you have to think about your own family history. You aren't biologically doomed by the ghosts of your ancestors' DNA, but you are absolutely heavily shaped by the environment your family created for you. Which leads us into what I found to be a deeply personal, almost unbelievable story from the source material. This is where Maarten's scientific theories crash into his actual life.
Yes, this is where the highly theoretical science of epigenetics becomes intensely, painfully personal for the guest. Maarten shares his own lived experience as what is known in the medical community as a DES son.
For those who don't know the history, and honestly, the original host Sonai didn't know either until he explained it, DES stands for diethylstilbestrol. It was a wildly powerful synthetic hormone, essentially a very early, very crude synthetic estrogen. And it was heavily, aggressively pushed by the global medical establishment back in the 1950s and 60s.
It was touted by pharmaceutical companies and doctors as an absolute miracle drug. The primary claim was that prescribing it to pregnant women would prevent miscarriages and ensure a healthy, robust pregnancy. Millions of women took it globally.
The tragic irony, of course, as we know now, is that it was later definitively proven not only to be completely ineffective at preventing miscarriages, but it also caused severe, devastating epigenetic side effects for the children who were exposed to it in the womb. It is one of the darkest chapters in modern pharmacology.
It is a textbook example of a massive, unintended epigenetic intervention. Endocrine disruptors like DES cross the placental barrier and completely scramble the delicate, perfectly timed chemical signals that tell a fetus how to develop its reproductive and nervous systems.
And Maarten's personal story with this is wild. His mother was 40 years old when she got pregnant with him. Now, prior to him, she had suffered a late-term miscarriage of a baby boy, which is obviously a profoundly traumatic event. Because of that medical history and her age when she got pregnant with Maarten, her doctors immediately prescribed her the supposed miracle drug, DES, to ensure she wouldn't lose another baby.
And she didn't tell him for decades. She carried that secret, or perhaps just the medical history, quietly for almost his entire life.
She didn't reveal this to him until she was 85 years old, and Maarten was 45. He was already a fully established DNA researcher, and the way he found out is almost comical if it weren't so serious. She finally wrote about her experience taking the drug in a personal essay she submitted to a writing contest for a popular Dutch women's magazine called Label. That is literally how he found out his mother took a massive synthetic hormone while pregnant with him.
Just imagine the cognitive dissonance of that moment. Imagine being 45 years old, an accomplished biologist who studies cellular structure for a living, and suddenly discovering that your own physical development in the womb was subjected to a powerful synthetic hormone with known severe epigenetic consequences. It must have been a staggering realization.
He talks about how all the puzzle pieces of his life suddenly clicked into place. The medical consequences for DES children were awful across the board. The daughters often developed rare vaginal cancers and severe fertility issues. The sons also faced reproductive abnormalities, but beyond the physical, there were profound, documented psychological effects. The source notes drastically increased rates of deep social anxiety, depression, and psychiatric disorders in the offspring. And Maarten says, reading that essay, suddenly his entire life made sense. He had suffered from severe, unexplained, crippling social anxiety his entire youth.
He describes his baseline childhood personality. He says he was naturally an optimistic kid, he had a good disposition, but he was incredibly withdrawn, dealing with this heavy, suffocating baseline of anxiety without having any environmental reason for it. He had good family, no obvious trauma, yet his nervous system was constantly in overdrive. It is a perfect, tragic example of a chemical altering the epigenetic landscape of a developing fetus, leaving a permanent, invisible mark on how their genes express stress and anxiety. The watering can, in his case, was a synthetic pharmaceutical.
But this revelation is exactly what triggered his entire pivot in life. It started his journey into what we now casually call biohacking, because his biological logic as a researcher was incredibly sound. He thought to himself, wait a minute, if one chemical substance, the synthetic estrogen from 1960, could alter my epigenetic landscape so profoundly and negatively, causing this lifelong anxiety, couldn't other substances or other daily habits alter it positively and reverse the damage?
It is the ultimate real-world application of the flexible landscape theory we discussed earlier. If the terrain was damaged, cratered, and made rigid by one environmental factor in the womb, can it be manually healed and reshaped by another environmental factor decades later?
So he just started experimenting on himself. He became his own primary test subject. And the source details some absolutely fascinating interventions he tried. First, he discovered running, just simple, hard cardiovascular exercise. He found that running a few times a week reduced his baseline lifelong anxiety by 30 to 40%.
Which, while simple, aligns perfectly with the most cutting-edge modern studies on exercise neuroplasticity. We know now that intense physical movement is a profoundly powerful epigenetic modifier. When you run, your muscles release proteins that travel to the brain and stimulate the production of BDNF, brain-derived neurotrophic factor. It's like miracle grow for your neurons. It literally changes how your DNA expresses itself in relation to stress tolerance. He went from being barely functional in some social situations to being highly functional, simply by changing his internal chemical environment through the physical stress of running.
Then he talks about something as simple as iodine. He started taking iodine supplements just because he read some research suggesting that many people in the modern world are chronically deficient. But he experienced a totally unexpected, completely bizarre side effect. For 50 straight years, he had a severe nail-biting habit. He would chew his nails down to the quick, a classic physical manifestation of chronic tension and deep anxiety. After just a few weeks of taking a daily iodine supplement, he looked down at his hands one day and realized he had fingernails. The lifelong habit had just stopped. The urge was gone.
Now, he is quick to acknowledge, as any good rigorous scientist would, that this is an N of one experiment. That means a sample size of one. In the scientific community, that's purely anecdotal. It could absolutely be a placebo effect.
Yeah.
But he insists he changed absolutely nothing else in his routine at that time. What's fascinating here is that the thyroid gland relies heavily on iodine to regulate hormones that directly impact the central nervous system. It highlights how highly targeted, seemingly minor nutritional changes can potentially ripple through our nervous system and alter long-standing behavioral outputs.
But the absolute craziest biohack he talks about, the one that really caught my attention, is EWOET, exercise with oxygen therapy. Have you ever heard of this?
I have, and it is a fairly extreme, highly targeted intervention. In the interview, Maarten carefully contrasts it with hyperbaric oxygen therapy. Hyperbaric chambers are often used now for treating things like long COVID or severe tissue damage, where you sit in a sealed tube pressurized at 2 or 3 times normal atmospheric pressure to force oxygen into the body. But that is incredibly expensive and requires a clinical setting, so his biohacking alternative is EWOTO.
Right, so here is what he does in his living room. He gets on a stationary exercise bike, and while he is doing a heavy, intense physical workout for 15 solid minutes, pushing his heart rate way up, he wears a specialized sealed mask that feeds him 95% pure oxygen from a massive bag.
To put exactly how extreme that is in perspective for you listening, the normal ambient air we breathe every day, the air you are breathing right now, is only about 21% oxygen. The rest is mostly nitrogen.
Exactly. So he is voluntarily flooding his entire cardiovascular system with 95% oxygen while his heart rate is absolutely pumping. What is the biological theory behind this?
The underlying theory, and again, this is in the biohacking realm, not standard medical advice, is all about blood plasma saturation. Under normal circumstances, your red blood cells carry oxygen. But when you create a massive demand for oxygen through heavy exercise and simultaneously supply a 95% pure source, you force the oxygen not just into the red blood cells, but deep into the blood plasma itself. The high cardiac output pushes this hyperoxygenated plasma deep into microscopic capillaries, reaching damaged tissues, reducing systemic inflammation, and theoretically reaching areas of the brain that normal resting breathing could never fully saturate. Maarten claims it is an incredibly effective, accessible hack for anti-aging your brain, especially after you cross the age of 50.
And the most tangible, immediate result he noticed from doing the CBOT protocol, a massive, undeniable increase in his ability to vividly remember his dreams every single night.
Which is incredibly telling. From a neurological standpoint, dream recall is heavily linked to the density and health of neural pathways in specific regions of the brain. If he is suddenly recalling dreams vividly, it suggests a significant shift in his brain's nocturnal activity and memory consolidation processes.
And this is where the contrast between the two speakers shines again, because the original host, Sana, hears about this vivid dream recall, and she immediately connects it to her own totally different experiences. She says she also remembers her dreams with intense, crystal clarity, but she achieves it by microdosing psychedelics. She notes that in her wellness circles, it's believed that psychedelics stimulate the release of natural DMT in the pineal gland, opening up that dream space.
It's two completely different paths, one mechanical and oxygen-based, one chemical and psychedelic, arriving at a similar neurological outcome. It raises a really important question for you to consider today. What small, or perhaps large, daily habits might be currently shaping your own epigenetic landscape? Are you actively feeding your landscape with sunlight, movement, and oxygen? Or are you starving it with chronic stress, poor nutrition, and stagnant routines?
It's such a wild thought to realize you are constantly programming yourself, which brings us perfectly into the next major theme of their conversation. We've touched on it, but let's dive deep. The source gets into psychedelics, the danger of biological rigidity, and the ultimate mystery of the cell, junk DNA.
The dynamic between the hosts really peaks here because Sonia is very open, very vulnerable about her use of psychedelics to explore the nature of reality and the human psyche. She talks passionately about having intense, mystical, ego-dissolving experiences that reveal the hidden depth and mystery of life to her. She views it as spiritual medicine.
And Martin's response to her spiritual passion is just so perfectly illustrative of his personality. He is a cutting-edge scientist. He is literally a biohacker who breathes pure oxygen on a bike. But he is also a self-proclaimed massive control freak. Remember, he rates his life a 10 out of 10 right now. He is happy. So he says he has absolutely no desire to lose his grip on reality or take a massive hero dose of psychedelics just to see what the universe looks like.
The risk-reward ratio just doesn't compute for him.
Exactly. He literally tells her that he has a specific plan. He is going to wait until he is 95 years old, when his body is failing, when he is sick, or when he simply has nothing left to lose. And then, he says, he is going 100% all in. He's going to take a massive dose and see where the trip takes him. The calculated risk management of planning a psychedelic trip 50 years in advance is just hilarious to me. But despite his personal reluctance, his rigorous scientific perspective on why psychedelics actually work for mental health is deeply fascinating.
It all ties perfectly back to the landscape analogy we started with. The source posits a compelling theory regarding severe, chronic trauma. We aren't talking about a bad day at work. We're talking about the kind of deep, therapy-resistant depression or PTSD that utterly ruins lives. Maarten suggests that when a human is subjected to a massive, prolonged traumatic stimulus, their normally flexible epigenetic landscape becomes rigid. It freezes. The chemical tags lock the DNA into a permanent hypervigilant threat mode, and the landscape loses its natural, healthy flexibility.
So the lush meadow and the shifting desert just turn into solid concrete.
Exactly. And because the flexibility of your DNA expression is directly physically coupled to the neuroplasticity of your brain also becomes functionally rigid. The neural pathways become deeply entrenched grooves. You get stuck in endless, agonizing negative thought loops because your physical biology has lost the ability to adapt and change paths.
So where do psychedelics like psilocybin from magic mushrooms actually come in biologically? Because they are showing incredible promise in modern clinical trials. Maarten explains them as a massive chemical disruptor. If the trauma turned the landscape to concrete, psilocybin is a chemical sledgehammer. He explains that these compounds radically, aggressively increase the neuroplasticity in the brain. They force neurons to grow new dendritic spines and form new connections almost immediately. And this forced neurological flexibility in turn forces that rigid, stuck epigenetic code to become fluid again. It literally shakes the entire biological system out of its frozen, traumatized state.
It is a profound mechanistic biological explanation for what many people describe as a mystical experience.
Yeah.
It explains why a single dose of psilocybin in a clinical setting can sometimes cure decades of severe depression, where traditional SSRI medications fail. They don't just temporarily numb the mind or boost serotonin. They may be literally unfreezing the physical epigenetic expression of the trauma.
Okay, that is mind-blowing. But let's pivot from the mysteries of the mind down to the physical, microscopic structure of the cell itself, because the source tackles what might be one of the biggest, most misunderstood mysteries in modern biology, the mystery of what we call junk DNA.
Yes. When the Human Genome Project was completed in the early 2000s, it revealed a genuinely shocking fact that confused scientists globally. Roughly 90 to 95% of human DNA does absolutely nothing. At least, it doesn't seem to code for anything. It doesn't make proteins. It doesn't build muscle tissue. It doesn't create enzymes. For a long time, the scientific consensus was simply to label this vast majority of our genetic material as junk, evolutionary leftovers.
But Maarten strongly, vehemently disagrees with that label. He points out that scientists are constantly, desperately searching these vast genetic deserts, looking for tiny hidden codes or secret sequences, but he thinks they are missing the point entirely by looking at it as a software code.
If we connect this to the bigger picture of cellular biology, Maarten views the DNA molecule not just as a linear information storage system, not just as a hard drive, but as a highly durable physical building material.
He even points out a wild fact. Bioengineers have literally built microscopic three-dimensional smiley faces out of synthesized DNA strands simply because the double helix is such an incredibly strong, reliable, programmable structural material.
His overarching theory is that the 90 to 95% of so-called junk DNA serves a vital, indispensable structural purpose. Think about the physics of a cell. The nucleus of a human cell is unimaginably small, yet you have to pack roughly 2 meters of DNA strand inside every single one. You can't just shove it in there randomly like a tangled ball of yarn. It has to be organized perfectly so the cell can read it. Maarten argues that the junk DNA acts as the physical scaffolding. It is the complex architecture that dictates exactly how the active, coding parts of the DNA fold, loop, and organize themselves inside that microscopic sphere.
So it physically creates the terrain. The junk DNA is the hills and valleys. Without the junk DNA folding everything in highly specific, mathematically perfect ways, the actual coding DNA wouldn't be positioned in the right place at the right time for those epigenetic watering cans to reach it. The environment couldn't interact with it. It is not junk at all. It is the vital 3D infrastructure of life.
It completely reframes how we must think about our genome. It isn't just a book of instructions. It is an architectural marvel of bioengineering.
It really is. Okay. As we move into the next major phase of their conversation, I need to pause the show for a second and issue a very clear, very strict reminder to you, our listener, regarding the content we are about to discuss. We are about to wade into some incredibly controversial waters. The source material features highly politically charged content regarding vaccines, specifically mRNA COVID-19 technology and global health interventions. Before we dig into his specific claims, I want to make it crystal clear we, as the hosts of this deep dive, are impartially reporting the views and statements expressed by the guest, Maarten Fornerod. We are not endorsing these viewpoints. We are not taking a political stance. We are simply conveying the biological arguments made in the original Wisdom podcast interview so we can analyze the source material provided to us. Let's look strictly at the science he's claiming.
That is an essential clarification, because Maarten Fornerod does not hold back. He expresses highly critical, deeply skeptical views on the global rollout of mRNA technology.
He really does. When Sana explicitly asks him if he considers the mRNA shots to be a form of genetic manipulation, he says, "We honestly don't know the bottom of it yet," but he issues severe, blunt warnings. He claims that the global rollout of these pharmaceutical products was essentially an uncontrolled, massive experiment. He specifically asserts that there was no adequate mid to long-term safety testing done. He claims there was no cancer research conducted, and crucially, no genotoxicity testing done before these completely novel therapeutics were administered to the global population, which included pregnant women and children.
Furthermore, moving into the socio-political realm, he claims that the emergency justification used for this unprecedented rapid rollout was artificially created and maintained. He argues this was achieved by the deliberate systemic suppression of alternative early treatment protocols by health authorities. He specifically mentions off-patent medications like ivermectin, hydroxychloroquine, and monoclonal antibodies, which he claims were highly effective but aggressively sidelined to protect the emergency use authorization of the mRNA products.
That is his stance on the politics and the medical bureaucracy of the pandemic. But let's look at the actual biological mechanism of his concern. As a DNA researcher, he breaks down exactly why he is so alarmed by mRNA technology inside the human body. And his argument centers on something called residual DNA.
Yes. Maarten explains that the manufacturing process for creating billions of doses of mRNA requires the use of DNA plasmids in giant vats. He claims that the final mRNA shots do not just contain the intended synthetic RNA, they also contain billions of fragmented pieces of DNA, which he describes as a residual byproduct, or a fundamental contamination, from that hurried manufacturing process.
Now, the original host, Sane, pushes back perfectly here. She asked the exact question I was thinking. But wait, DNA is literally everywhere. We eat DNA every day in our vegetables, in meat, in caviar. Why is swallowing DNA fine, but this residual DNA dangerous? And Maarten's answer focuses entirely on the microscopic delivery mechanism used in the shots, lipid nanoparticles.
This is the absolute crux of his biological argument against the technology. He explains the difference in exposure. If you eat DNA in a salad, your highly acidic digestive system rips it apart immediately. If naked, unprotected DNA somehow gets directly into your bloodstream, your immune system enzymes recognize it as foreign debris and quickly shred it. But, he explains, the residual DNA fragments inside these shots are encased in lipid nanoparticles. These are tiny, highly engineered synthetic fat bubbles designed specifically to bypass the body's natural defenses, evade the immune system, and fuse with cell membranes to deliver their payload inside.
And his major concern, the thing that alarms him most, is that these lipid nanoparticles do not just stay localized in the deltoid muscle of your arm where the needle goes in. He argues that the moment they hit the bloodstream or the lymphatic system, they travel systemically throughout the entire body. Because their sole function is to penetrate cells, he claims these lipid nanoparticles, carrying both the mRNA and the contaminating residual DNA fragments, penetrate cells indiscriminately. They go into heart tissue, liver tissue, brain tissue, ovaries, everywhere.
And once that payload is delivered inside the cell nucleus, his ultimate fear is genomic integration. He worries that these foreign, synthetic DNA fragments could physically splice and integrate into the host human's own permanent DNA genome. If that happens, you alter the hard code. He states that the frequency of this happening, whether it occurs in 1 in 1,000 cells or 1 in 1,000,000 cells, is currently unknown because the proper genotoxicity studies weren't mandated, and it is an active, urgent area of independent research he is currently involved in.
He plainly expects that this systemic delivery mechanism will cause significant DNA damage over time, and he states on the record it would highly surprise him if it didn't. When Sinai asks why this isn't front-page news, he attributes the lack of mainstream scientific focus on this risk to what he calls the dogmas of the modern medical establishment. He argues that major science and health institutions have become huge, bureaucratic, top-down organizations run by financial managers and politicians, not actual curious scientists, creating a rigid dogma that actively suppresses dissenting research or inconvenient questions.
This deep skepticism of top-down authority leads directly into another major controversial assertion he makes. He claims that the incredibly rapid development of these mRNA platforms was fundamentally a military-driven project, heavily funded and guided by the military-industrial complex in the United States, rather than a standard altruistic public health initiative.
And he uses this military connection to introduce a terrifying but very real concept, dual-use technology.
Dual-use technology is a term used to describe innovations or research developed ostensibly for military defense that inherently, inescapably possess the exact capability for offensive use. In this specific context, Maarten argues that a military biodefense base might pour billions into developing rapid mRNA vaccine technology as a defense to quickly protect soldiers against a novel biological weapon.
But, Maarten warns, it's a double-edged sword. If you possess the incredibly advanced technology and the highly guarded, secretive facilities required to rapidly manipulate biology to create a customized defense, you inherently possess the exact same technology to create the bioweapon itself, to engineer a highly lethal, customized virus from scratch. He argues that military organizations globally, not just in the US, are absolutely engaged in this kind of gain-of-function research. He views manipulating complex biology at this foundational level as incredibly reckless, given the massive historical potential for unintended consequences, lab leaks, or pure human error.
He extends this severe warning against playing God with complex biology to other modern interventions as well. Sinai asks him for other examples of this biological hubris, and he immediately brings up the heavily publicized project involving genetically modified mosquitoes.
Yes, the project heavily backed by figures like Bill Gates and various global health foundations. The premise sounds amazing. They genetically modify millions of male mosquitoes in a lab so that when they mate in the wild, their female offspring die before they can bite. They release these modified mosquitoes into the wild with the noble goal of crashing the local mosquito population to eradicate malaria and dengue fever.
It sounds like a technological triumph. But Maarten calls this approach sheer madness. His argument is purely ecological. He states that human beings cannot possibly model or predict how a complex, highly integrated biological ecosystem will react to a massive artificial genetic intervention. What happens to the food chain that relies on mosquito larvae? Will the modified mosquitoes mutate in the wild? Will they crossbreed in unexpected, catastrophic ways?
He compares it to a famous historical ecological disaster to make his point, the intentional introduction of the massive Nile perch into Lake Victoria in Africa back in the 1950s. The idea back then was purely economic, to boost the local commercial fishing industry by introducing a huge, meaty fish.
But the hubris of the intervention ignored the complexity of the ecosystem. The massive Nile perch ended up eating virtually all the other native cichlid fish. It utterly decimated the existing delicate biotope, destroyed the entire natural ecological balance of the largest lake in Africa, and ironically ended up ruining the livelihoods of the local traditional fishermen. Maarten views the genetic mosquito project as the exact same kind of blind, top-down hubris, just on a genetic level rather than a macroscopic level.
He also briefly points to the recent reintroduction of wolves in the densely populated Netherlands as another example of a romantic biological experiment with severely unforeseen, chaotic consequences for local farmers and ecosystems. And he absolutely doesn't spare the pharmaceutical industry's latest darling from his critique either, Ozempic.
Right. Ozempic and the whole class of GLP-1 agonist drugs are currently being hailed as a miraculous, effortless cure for the global obesity epidemic. People are taking it just to lose a few stubborn pounds. But the original host, Sana, mentions a deeply unsettling anecdote. She talks about having a guest on a previous show who had just started taking it, and the guest was sweating profusely, shaking, and having severe heart palpitations right there in the recording studio.
Maarten nods along to this and points out that while the drug undeniably causes massive weight loss, the mechanism is brutal. It works by essentially paralyzing the movement of your stomach so food doesn't empty, keeping you artificially full so you literally can't eat. He warns that when you artificially hack the body's deeply evolved metabolic and endocrine systems so drastically, severe side effects are guaranteed. He mentions the physical symptoms, like the severe nausea and heart palpitations, but he also issues a stark warning about the significant, often underreported psychological changes and depressive episodes that occur when you forcefully alter the gut-brain axis with synthetic peptides.
So synthesizing Maarten's entire overarching philosophy from this segment, he is urging profound, relentless skepticism toward any top-down miracle interventions. Whether they come from a pharmaceutical giant pushing a lucrative weight loss pen, a well-meaning tech billionaire trying to alter mosquitoes, or military-backed global genetic health mandates. His fundamental belief, born from decades of studying DNA, is that the vast fractal complexity of biology will almost always outsmart our crude attempts to forcefully, artificially control it.
It is a sobering, slightly terrifying perspective on modern science and medicine, but it also brings us to the crucial, empowering final phase of our deep dive today. We've talked about the dangers of artificial interventions, but what is the practical, natural application of all this knowledge? So what does this all mean for you? How do we take care of our own epigenetic landscape safely, naturally, and effectively?
To answer that, Maarten points to what I think is the most hopeful, inspiring part of the entire interview. He describes an experiment that serves as the ultimate proof of epigenetic power over genetic destiny. He talks about a very specific, famous strain of laboratory mice often used in research. These mice have been heavily genetically bred over generations to be incredibly, predictably unhealthy. They possess a hard DNA code that practically guarantees they will develop early-onset cancer, severe obesity, and fatal heart disease. Genetically speaking, they are entirely doomed from birth.
But here's the profound, beautiful twist that changes everything. When researchers take these genetically doomed mice and intervene purely with their environment, specifically by placing them on a strictly healthy, highly optimized, nutrient-dense diet from birth, something miraculous happens in their biology.
They do not get early cancer. They do not develop severe obesity or heart disease. They end up living completely normal, healthy, vital lifespans, completely identical to control mice that never had those genetic defects in the first place.
Their healthy environment completely, entirely overrides their bad genetic code. The watering can of good nutrition and proper environment turns the genetic desert of inevitable disease into a thriving meadow of health. It is the ultimate proof of concept.
It proves, fundamentally, that your hard DNA is not your absolute doom. It is merely a set of biological tendencies and predispositions that your daily environment can either aggressively activate or successfully suppress. Based on this, Maarten provides a very tight, highly practical list of actionable tips for clearing your own epigenetic baggage and optimizing your landscape.
Let's run through his protocol. First on the list, heavily based on his own life-changing experience with curing his lifelong social anxiety, exercise. But not just for fitness. Physical movement acts as a massive systemic chemical signal to your epigenetics that you are capable, that you are adapting, and that you are resilient. It floods the system with those neurotrophic factors we discussed.
Second on his list, prioritizing high-quality, uninterrupted sleep. We often think of sleep as just resting, but biologically, the brain and the body use the deep stages of sleep to physically repair the epigenetic landscape. It is when the cellular maintenance crew comes in to fix the damage done by the stress of the day. If you cut sleep short, the repair doesn't happen, and the damage compounds.
Which leads directly and unavoidably to his third point, eliminating alcohol. He states very plainly, without judgment, but as a biological fact, that drinking alcohol is fundamentally unwise for your biology because it utterly ruins your sleep architecture. It prevents you from reaching the deep REM states required for that crucial epigenetic maintenance. You might fall asleep faster after a drink, but your brain isn't repairing the landscape. And finally, he advocates heavily for practices that actively maintain mental and therefore epigenetic flexibility. He highly recommends daily meditation to train the nervous system to quickly return to a baseline of calm rather than getting stuck in a rigid threat state. And, as we discussed in depth, for some people under the right clinical or guided circumstances, the careful, intentional use of psychedelics to violently break out of rigid, trauma-induced epigenetic states and restore neuroplasticity.
It all comes down to being a proactive participant in your own body. Try a keto diet and see how you feel. Try running, commit to meditation, change the inputs, and you will inevitably change the landscape.
It is a deeply, profoundly empowering message to end on. You are the active gardener of your own epigenetic landscape. You may not have chosen the specific plot of land you were given at birth, your hard genetics, but you have immense daily power over what grows there.
As we wrap up this incredible, wide-ranging journey today, from the grassy meadows and hidden deserts of our epigenetics to the architectural mysteries of junk DNA, all the way to the controversial frontiers of biohacking and the massive implications of modern mRNA medicine, I want to leave you with one final, truly mind-bending detail from the source material.
It is a thought that really forces you to step back from the microscope and look at the absolute grand cosmic scale of life on Earth.
Toward the end of the interview, Maarten points out that the hard code of DNA, that specific double helix language built from 4 chemical bases, is universal across almost all life on this planet. A microscopic bacteria swimming in a puddle, a massive elephant on the savannah, the oak tree in your backyard, and you sitting here listening to this right now, we all run on the exact same fundamental biological operating system. And scientists estimate that this incredibly complex, perfect code originated roughly 2 billion years ago.
2 billion years. And because there are obviously no biological fossils that old with intact organic material, we have absolutely no scientific idea where that universal code came from, how it originally formed in the primordial soup, or who or what wrote it. It is the greatest mystery in science.
So here is a final provocative thought for you to ponder long after this deep dive is over. We spend so much of our time worrying about what our DNA dictates for our daily lives. We stress over inheriting family traits, we worry about genetic risks for disease, we obsess over our weight and our height. But maybe, occasionally, we should zoom way out and ask ourselves a bigger question. What does it truly mean to be the temporary walking conscious custodians of a mysterious 2 billion-year-old universal code? You are carrying a literal piece of the universe's oldest, greatest mystery inside every single one of your trillions of cells.
It really changes your perspective on what a human life actually is, doesn't it?
Yeah.
It elevates the whole human experience.
It really does. I want to close today with a beautiful Chinese proverb that Martin, honoring his own mixed Indonesian and Chinese heritage, shared with Sanai at the very end of their interview. Give more than you receive, and you will receive more than you give. Thank you so much for joining us on this deep dive today. Keep tending to your landscape, keep questioning the world around you, and we will catch you next time.